welcome to the third meeting of Food Biotechnology class. Still with me, Pujiati, the lecturer of Biology Education Department, Universitas PGRI Madiun, Indonesia. In this meeting, I will discuss about the most popular traditional fermented food from Indonesia that is called TEMPEH. So in this part, I will talk about some topics about TEMPEH. The first one is the definition of TEMPEH, the history of TEMPEH, the types, the production, the nutritional value, the variation food of TEMPEH, and the last one, the bioactivity of TEMPEH. Okay, the first topic is about the definition of TEMPEH. TEMPEH is a traditional Indonesian soy product that is made from fermented soybeans. It is made by a natural culturing and controlled fermentation process that binds soybean into a cake form. Here, a special fungus is used, that is Rhizopus oligosporus, usually marketed under the name as TEMPEH starter, or in Indonesia we call it ragi tempe. Like tofu, TEMPEH is made from soybeans, but it is a whole soybeans product with different nutritional characteristics and texture qualities. TEMPEH's fermentation process and its retention of the whole bean give it a higher content of protein, dietary fiber, and vitamins. It has a warm texture and an earthy flavor. TEMPEH is known to contain vitamin B12, which is essential for a healthy human diet. Therefore, TEMPEH is a particular interest of vegan diet since B12 is normally found only in the animal-derived products. Next, we talk about the history of TEMPEH. TEMPEH originated precisely Central or East Java, with an estimated discovery between a few centuries ago to 1,000 years or more. Around the 12th to 13th century, a type of food was mentioned as kadele in an old Javanese manuscript named Serat Sri Tanjung. However, it is not clear whether kadele referred to processed fermented soy or not, since the term in Javanese today refers to soybeans. The earliest known reference to it as TEMPEH appeared in 1815 in the Serat Centini. The invention of TEMPEH is connected to tofu production in Java. The tofu making industry was introduced to Java by Chinese immigrants around the 17th century. Chinese-Indonesian historian Ong Hock Ham suggests that TEMPEH was accidentally produced as the byproduct of the tofu industry in Java, as discarded soybeans caught the spores of the various fungus that was found to be edible. Three detailed, fully documented stories of TEMPEH worldwide have been written all by Shortleaf and Aoyogi in 1985 and 1989 and 2001. Okay, let's talk about the types of TEMPEH. The most common TEMPEH is made from fermented soybeans. However, traditionally, other ingredients such as ampas tahu or tofu drag, ampas kelapa or coconut drags, and peanut may be used in a fashion similar to the TEMPEH making process, although perhaps using different fungus or attracting other microbes. Okay, the first kind of TEMPEH is TEMPEH kedelai, also called with TEMPEH kedelai or TEMPEH delai or simply just TEMPEH. The most common and widely known TEMPEH made from controlled fermentation of soybeans. This is the picture of TEMPEH kedelai. Next type is TEMPEH gembus, soft and fluffy TEMPEH made from soy pulp or tofu dregs. It is known by different names across Java, for example as tahu cokol or tahu susur in Temanggung. This is the picture of TEMPEH gembus. And third one is oncom. TEMPEH oncom or simply oncom is made from peanut pressed cake or soy dregs. Among fermented bean products, oncom is more prevalent in West Java, where it serves as the main ingredient in various Sundanese cuisines. There are two types of oncom, a bright red-orange kind with Neurospora cytophila and the black one with the same fungus as TEMPEH uses. And the next is TEMPEH menjes kacang, a specialty of Malang. A rough textured TEMPEH menjes is made from black soybean mixed with other ingredients such as peanut dregs, cassava fibers, and soybean meal. The process of making menjes kacang is quite similar to black oncom. The fifth type is TEMPEH bongkrek. TEMPEH bongkrek is a variety of tempeh from Central Java, notably Banyumas Regency, that is prepared with coconut dregs. This type of tempeh has led to several cases of fatal food poisoning, as it occasionally gets contaminated with the bacterium Burkholderia gladioli, and the unwanted organism produces toxins named bongkrek acid and toxoflavin from the coconut, besides killing off the rhizopus fungus due to the antibiotic activity of bongkrek acid. Fatalities from contaminated TEMPEH bongkrek were once common in an area where it was produced. Thus, its sale is now prohibited by law. Clandestine manufacture continues, however, due to the popular flavor. The problem of contamination is not encountered with bean and grain tempeh, which have a different composition of fatty acid that is not favorable for the growth of P. gladioli, but encourages growth of rhizopus instead. When bean or grain tempeh has the proper color, texture, and smell, it is a very strong indication the product is safe. Yellow TEMPEH bongkrek is always highly toxic due to toxoflavin, but TEMPEH bongkrek with a normal coloration may still contain lethal amounts of bongkrek acid. Okay, the next one is oat tempeh. A new form of tempeh based on barley and oats instead of soy was developed by scientists of the Swedish Department of Food Science in 2008. It can be produced in climatic regions where it is not possible to grow soybeans. The last type is TEMPEH semangit. In Indonesia, ripe tempeh, 2 or more days old, is considered a delicacy. It is also known as TEMPEH semangit or stinky tempeh in Java. Having a slightly pungent aroma, small amounts are used as a flavoring agent in a traditional Javanese dishes. This is the picture of TEMPEH semangit. So, this is a table based on Erkan et al. Research. The table is described about the proximate composition and physicochemical properties of the tempeh from several samples, and also the organic acid composition of the tempeh also from the several samples. Okay, let's move to the next topic about the tempeh production. This is the picture of the tempeh production process. First, soybeans as the raw material boiled, then soaked around 36 hours. Second, peel the husk and clean it. Third, steam around 30 minutes and liquid. Fourth, give a starter culture or ragi estimated 1%. 5. Cover with banana leaf or plastic and ferment around 48 hours. And the last, the tempeh is ready to consume. And this is the flow diagram of the tempeh preparation process by Knott and Kish Research 2004. Inside the production process we mentioned previously, there is the soaking stage. The microflora of soybean tempeh is complex and its development starts during the soaking of the raw ingredients. The microbial composition of traditional tempeh is determined by ecological factors such as the acidification by lactic acid bacteria during the soaking stage, the lethal effect of the cooking operation, contamination caused by handling during cooling, the composition and vitality of the inoculum, heat and mass transfer limitation during the fungal fermentation, the incubation condition, and the condition under which the product is stored. Fresh tempeh contain high number of mesophilic aerobic bacteria as well as enterobacteria, staphylococcus, and yeast. In addition, psychotropes may develop during refrigerated storage. Lactic acid bacteria dominate during the soaking stage of the traditional process. As a result, a significant increase of organic acids takes place, as you can see in Table 1. Acidification and other inhibitory effects of lactic acid bacteria were also shown to suppress the natural microflora, such as coliform, Klebsiella pneumoniae, and yeast, and to improve the shelf-life of tempeh. However, because of climatic and processing difference, this lactate fermentation does not occur by itself in temperate climates. Solid-state fungal fermentation occurs in this process. Several hours after the inoculation of the beans, the germination of fungal sporangiosperms starts, followed by the development of mycelium. The mycelium penetrates into several layers of soybean cells, up to 25% of width of the cotyledons. Similar observations were made in a microscopical study of the growth of Rhizopus oligosporus on quinoa seeds, which first had to be decorticated or cracked in order to make them accessible. The entanglement of hyphae results in a certain cohesion of the fermented mass. As the firmness of tempeh is an important quality attribute, measurement of the strength of tempeh is appropriate for quality assessment purposes, but it provides little information about the amount of fungal biomass formed. Okay, next go to the nutritional value of tempeh. The soy carbohydrate in tempeh become more digestible as a result of the fermentation process. In particular, the oligosaccharides associated with gas and indigestion are greatly reduced by the Rhizopus culture. In traditional tempeh making shops, the starter culture often contains beneficial bacteria that produce vitamins such as B12, though it is uncertain whether this B12 is always present in bioavailable. In Western countries, it is more common to use a pure culture containing only Rhizopus oligosporus, which makes very little B12 and could be missing Citrobacter frendi and Klebsiella pneumoniae, which have been shown to produce significant levels of B12 analogs in tempeh when present. Whether these analogs are true, bioavailable of B12 has not been thoroughly studied yet. The fermentation process also reduces phytic acid in soy, which in turn allows the body to absorb the minerals that soy provided. So this is the nutritional value based on the USDA database. You can see here. The production of tempeh always involves tempeh starter or starter culture, which is made from microorganisms. The major genus of importance for tempeh making is Rhizopus microsporus with varieties Microsporus, Oligosporus, Rhizopodiformis, and sinensis, based on North and Ramboth's 1990. This is the picture, the starter culture of tempeh. The first picture shows the most popular starter culture in Indonesia, called ragi tempeh. The next topic is variation of food tempeh. There are variations of tempeh such as salad, crepes, steak, burger, juice, mendoan, nugget, and satay. We have discussed about the definition, the history, the fermentation process, and also the variation of foods which are made from tempeh. Next, let's move to discuss the bioactivity of tempeh from several studies. The first one, it is about the antioxidant activity of tempeh. Tempeh exhibits enhanced antioxidant activity compared to soybeans. For examples, tempeh extracts better scavenge DPPH, free radical, and superoxides over their unfermented counterparts. The improved antioxidant effect of tempeh may be attributed to increased level of polyphenols released from soy due to the soaking and boiling treatments, coupled with cell wall degradation by enzymes secreted by Rhizopus oligosporus during fermentation. Although fermentation initially decreased total isoflavone levels, in tempeh, biotransformation of isoflavonoid glycans, especially diazine and genisteins. Genistein and diazine levels doubles in tempeh compared to levels measured in unfermented soybeans. The second one is about the antimicrobial activity and effect on intestinal microbiota. In addition to antioxidation, tempeh exhibits antimicrobial activity and may also have the ability to modulate intestinal microbiota composition. Metabolites produced by tempeh starters, especially by those using Rhizopus strains, have shown in vitro antimicrobial activity against both gram-positive and gram-negative microorganisms. In tempeh, bacteria are not associated with the usual starter have been shown to possess antimicrobial activities. In another study, tempeh digested by simulated human gastrointestinal juices was then incubated with sample of human fecal bacteria, and increased number of bifidobacterium and lactobacillus compared to unfermented soy were observed, leading to conclude that component of tempeh modified through gut transit might contribute to its impact on the intestinal microbiota. This is a summary of functional activities associated with tempeh based on some studies. Okay, the last one, I will talk about the peptide bioactive from tempeh based on Tamam study. Studies on the bioactive peptide in tempeh are rare. Fermentation allows microorganisms to secrete proteolytic enzymes capable of converting the protein in soybean into peptides, such as dipeptide, tripeptide, and oligopeptide, possessing many biofunctional properties. Bioactive peptides are usually encrypted in their storage protein and are commonly composed of 2 to 20 amino acid residues. These peptides are functionally active after protein cleavage by proteolytic enzymes during fermentation. The conversion of the storage protein into bioactive peptides occurs through 2 major mechanisms, for example, fermentation and enzymatic hydrolysis. So everyone, that is the explanation about the tempeh. Thank you and see you on the next session.